Hello, everyone. Hello, people. So, guys, how are you? Welcome to the NEET Bytes session by Meritnation once again. Good evening, Paras. Good evening, Rebecca. Meghna, good evening. Hello, Saurabh. Very fine. Very good. So, hello, Noshin also. Hello, Hariharan. Ankit, welcome. Good evening to all the people out there. Okay. Good evening. A very good evening. Good evening to all the puns. All right. So another session of your NEET Bite happens to be on anatomy of flowering plant. Another very interesting chapters. Okay. And this session will be taken by me. I am Gausia. I shall be taking you through the different concepts of anatomy. For that matter, we are going to see different questions that have come. Okay, what are the concepts you can learn from there? Okay, and what is the pattern that is followed for this particular topic? Okay, of course, Ankit. Of course, that is the main issue here. Okay, this chapter appears to be confusing for the reason that you talk about different types of tissues present in the plant. Okay, and sometimes learning them becomes a task. However, we are going to see some questions where you find that concepts, if you keep them in mind, then it's not difficult to solve those questions. Okay, so definitely we are going to talk about meristematic tissues, right? We are going to talk about other tissues as well. Okay, so let's get started, everyone. All right. Okay, now before we begin with the anatomy section of this particular topic today, there was a question in previous video. Okay, if you were present in the previous session of morphology, okay, that was regarding the andrecium of hibiscus flower. Okay, that was regarding this particular flower. Okay, and as a matter of fact, the explanation was given right. However, the option chosen was wrong. Okay, so in this particular question, which is from the morphology, the answer is monadelphous, because in China rose, what happens? What do we find? We find that filaments, okay, filaments are all united. Okay, they are all united in one bunch. Okay, this is what you find. Okay, so that is why monadelphous is the right answer. This is just a topic from the previous session on your morphology. Okay, exactly. So you are getting it right, everyone, right? So very good, very fine. Okay, now starting with today's topic. Okay, let's get started with today's topic. And let me just okay, so that it's now visible to all of you. Fine. So today we will be discussing about plant anatomy. Right. So what basically is anatomy? Okay, what basically is anatomy? It is about the study of, this is about the study of internal structures. Okay, if you are looking into the internal structures of an organism, that then you say that you are looking into the anatomy of that particular organism. Okay, and for studying into the internal details of the body, okay, for studying into the internal topics, then we need to do the dissection for that matter. Okay, we need to perform dissection. Okay, so wherever you come across the word anatomy, what you have to keep in mind, that we are dissecting particular organism, and then we are studying into the internal details of the body. Okay, this is the idea about anatomy. Okay. Now, if you talk about the internal details of plant, then there are different types of tissues that you come across. Okay, there are different organizations that you come across, and so we have classified tissues of plant on the basis of their differentiation. Okay, we have classified them on the basis of differentiation. What does it mean? What is the meaning of classifying something on the basis of differentiation? Anyone can answer this question? What is the meaning of classifying something on the basis of differentiation? Yes, anyone can answer this particular question? Yes, I'm waiting for your answers. Can anyone tell me what is the meaning of, you know, considering differentiation? Yes, okay. They differentiate to form different structure. One answer I've gotten from Minakshi. Okay. Composition, right? Okay. Differs in the physiology. Perfect, Nivea. Okay. So basically, in plants, we find that there are certain cells who continue to divide forever. Okay. They never get their functions assigned. Okay, exactly. They never get their function assigned, and their task is to keep on dividing and contributing in the formation of more and more cells. Okay, this is what some of them are doing. The rest of the other cells, which are coming from such cells, are getting differentiated in the sense that their function is becoming defined. Okay, their function is becoming defined, right? Okay, so that is why we say that we have two types of tissues in plants, right? Yes, Krisha, welcome. I can see you. Yes, Krisha. Hi, Anjali, welcome. No problem. Okay, so we have some meristematic tissues. Okay, we call them meristematic tissues, and then permanent, in the sense that permanent tissues lose their power of division. Okay, and they become fixed for a particular function. Meristematic tissues continue to divide, and they continue to add into the mass of the plant body. Okay, this is the idea behind classifying the tissues of plants into meristematic and permanent. Okay, exactly, Rebecca. So meristematic tissues are capable of division throughout. Okay, then you can further classify the meristematic tissues on the basis of their origin. Okay, if you're classifying them on the basis of their origin, you can classify them on the basis of their place where they are present. Okay, their location, right? We can classify them like that. So we can have some pro-meristem, we can have primary meristem, we can have secondary meristem. Okay, on the basis of location, right, we can have apical meristem. Okay, intercalary and lateral meristem. Okay, fine. And then coming onto the permanent tissues, here also we can have two broad categories, being simple, made up of same types of cells, complex, made up of different types of cells. Okay, that is the broad classification we follow in case of plant tissues. Okay, exactly, Ankit, very fine. Simple can be further of parenchyma type. Okay, you've heard about this name since long, since your journey with your biology. Okay, since you've entered into the uh, world of biology. Then collenchyma. Okay, Sclerenchyma. So these are certain names that keep on coming every now and then. Okay, you know them since beginning. You happen to know somehow. Okay, you talk about them very often, isn't it? Okay, in complex we have xylem and phloem. Right. So now what do? We are going to go to the questions now. Okay, we will enter into the questions. We'll try to find out what are the type of questions we get from these particular things that we have seen. Okay, exactly, Minakshi. From your ninth standard, you are hearing about them. You are talking about them. Okay, very fine. Right. So, are you guys ready for the first question? Shall we begin with the questions, everyone? Are you guys ready? Yes? Okay. Very fine. Very fine. So, moving on. Moving on to the very first question. Okay. So, here we have the first question. Okay. It is an AIPMT question. And this question is uh, taken as first in the sense that it deals with roots. Okay. It deals with roots, right? Okay. So, the question is asking you that if you have the longitudinal section, okay, if you are having a LS, a longitudinal section of a root, then starting from the tip to upwards, there are 4 zones present. Okay. Which of the following is the correct order? Okay. So I'm already getting certain answers. Now, before we move into the answer, before we talk about the options, you all must understand that in case of root, okay, in case of root, the apical meristem, okay, the apical meristem is not present at the tip. Okay. It is not at the tip. Can you tell me why it is not at the tip in case of root? Root apical meristem, basically, if I talk about. Okay. Can you tell me why is it not at the tip? Anyone? Can you tell me why it is not at the tip? Right? Anyone? Any idea? Why am I saying like that? That apical meristem, however, I'm calling it as apical, but I'm saying it is not present at the tip. So what is wrong over here? Why is it not occupying the tip position? Exactly, it is protected. Very fine. Protected by what? Meenakshi and Rebecca. That's right answer. But what is the thing which is protecting it? Okay, fine. I'm getting this particular thing. It is because of the protection issue. But who is protecting it? What is that particular structure? Arsalan? Right answer. Okay. So at the tip, what do you have? You have root cap over there. Okay. You have gotten root cap over there. So root cap becomes apical, and root apical meristem, for that matter, becomes subapical. Okay, it becomes subapical in position. Right? Okay. So basically, when you talk about the different zones, okay, when you talk about the different zones of the roots that you come across, so first thing happens to be root cap. So root cap is here as well, root cap is here as well. So either option A is right or option D is right. Okay, either option A is right or D is right, because now we know that root cap is apical. Okay. Then seriously, what happens right after the root cap, as you guys are saying, that apical meristem has to be protected. So it implies that right after the root cap, who is following? Meristem is following. Okay, root meristem is following, which is why we say that cell division, the zone of cell division, will be the second most region. Okay, so again, in option A and D, we are having cell division as the second option. So again, either option A is right or option D is right. Right? Okay, okay, very fine. Now comes the deciding factor. Okay, now comes the deciding factor. So in order to get to the deciding factor, you must know that there are root hair present. Isn't it, people? Guys, you know about the root hairs? Right? There are root hairs present. Okay, I'm just coming on that, Priya. Coming on that. So can I know from which region root hairs are arising? Right, Shreya? Okay, so root hair are arising from the uppermost portion. Okay, they are arising from the region of cell maturation. Okay, hence we got to know an idea that when you are looking into a root, okay, when you are looking into a root, you find root hairs are coming from the topmost position. That is why cell maturation has to be the fourth most zone. Okay, the topmost, and in between you are going to have the cell enlargement. So option D appears to be the right option. So all the people who have given the right answer, congratulations. It was a correct answer. All the people who gave D as the answer, very good. Okay. Enlargement can also be called as elongation. Okay. That's not a that's a just a synonym here. Okay. It is known as elongation as well. Okay. You can call it as cell enlargement, alternatively cell elongation. Very fine. So is that clear to all the people? Clear, everyone? Perfect. Okay. Moving on. Next question. Going to the next question. Okay. Now this is an easy one. Okay, this question is an easy one. It is kind of a revision of some of the topics you've done already. Okay, so which element do you think is necessary for middle lamella? Okay, this is very easy question. I am sure I'll be getting floods of right answers here. Okay, there must be a flood of right answers, everyone. Yeah, it has started. Very good. So Minakshi, Meghna, and Arsalan, perfect. It is calcium which is required. Okay, that's right. Can anyone tell me what is the other thing you need to have? Pectin. Okay, Rebecca, very good. So it is basically present in the form of calcium pectate. Very nice. So all of you, right answer. Keep going good. Okay, this was an easy question, right? Moving on to the next one. Next one. Okay, this is a good question. Talks about two important people in the plant body. Okay, talks about xylem and phloem, right? And why do I call them important people? Because they are responsible for carrying things to and fro. Okay, both xylem and phloem, right? So question is asking you what happens during vascularization in plants. So what is the meaning of vascularization? What is the meaning of vascularization? Can you guess? Any guessing work over here? Yeah, what is the meaning of vascularization? Right? I'm waiting. Okay. Formation of xylem and phloem, right? Okay, because they are in the present in the form of vascular bundles. Okay. And why do we call them as vascular bundles? Because they are responsible for conduction. Okay, they are responsible for the conduction of food and water, right? Conduction of materials. Very fine. That is why we call them as vascular bundles, vascular tissues, right? So what happens during vascularization? Okay. Now you know that we call that particular meristem as cambium, okay, procambium for that matter, which is responsible for forming xylem and phloem. So let's read the options one by one. Let's try to find out which option is matching the right theory over here. Okay. First option says that differentiation of procambium, okay, followed by primary phloem and then primary xylem. Now here is something wrong. Okay, phloem doesn't proceed. It doesn't precede the formation of xylem. Okay, xylem formation has to be first. Okay, it has to happen before the phloem. So this option is wrong. Moving on to the B1. Differentiation of procambium, xylem, phloem simultaneously cannot happen. Okay, why? Because procambium must mature first. Procambium is meristem over here. Okay, so procambium must mature first, then you are going to have xylem and phloem, right? So again, this is a wrong option. Okay, next one. Differentiation of procambium, cool, followed immediately by the development of secondary xylem and phloem. Now there is a problem here. Okay, there's a problem because secondary xylem and phloem do not come from the procambium. They come from vascular cambium. Okay, they come from the vascular cambium, that is also called as secondary meristem, also known as lateral meristem, and we know it is responsible for the secondary growth in plants. Okay, so procambium is not leading to the secondary growth. Hence, secondary word over here is making this option wrong. Okay, let's see the last one. Differentiation of procambium. Okay, cool. Followed by development of xylem and then phloem. Okay, so this is the most appropriate option we are having. So the correct answer is D. So Shreya, Kumari, Meghna, Dimple, and Smriti, congratulations, congo to all the people who've given the answer as D. Okay, you guys are going good. Very fine. Understandable, this thing, everyone? This thing is understandable? So we've seen all the options one by one. We've tried to find out why is this right and the others are wrong. Okay, right. Moving on to the next question. Okay, very fine, very good. So here comes your next question. Okay, this is again an AIPMT question, and it talks about again very important details. Okay, a common structural feature of vessel elements and sieve tube element. See everyone, the question is asking you about the commonality. Okay, you have to talk about the common thing, that common feature that you find in the xylem and in the sieve tube. Now you tell me one thing, sieve tubes and vessel elements, which one is belonging to whom? Okay, vessel elements belong to xylem or phloem. Hello Alia. Hello Bandana. Vessel elements belong to xylem or phloem. First, you answer me this thing. This is just an oral test going on. Question within a question. Okay. So it belongs to xylem. I got an answer from Rebecca. Okay, belongs to xylem. What about sieve tube elements, guys? Okay, Arslan, that's right. Smriti, cool. Dimple, cool. Very good. What about the next one? Sieve tube. Very good, Ankit and Ishita as well. Okay. This one, guys. This one belongs to phloem. Perfect. Okay. This one belongs to phloem. Okay. Now we have to find out something which is common in between both of them. Okay. So we will again go one by one. Okay. We'll see each option one by one. Now, first option says thick secondary walls. Now there's a problem here. The problem is that vessels do have secondary walls. Okay, they do have thick secondary walls. Okay, they do have thick secondary walls present, but this is not true for sieve tube elements. Okay, sieve tube elements do not have the thickenings of secondary wall. Reason because they show us the presence of plasmodesmata. If you guys remember a little bit of detail, then you must remember that your sieve tube elements, let's say this is one sieve tube element, it is connected with a companion cell. Companion cell is companion because it is always accompanying the sieve tube, and both of them are showing us the plasmodesmatal connection. Understand, right? That is why it doesn't have the secondary thickenings. It doesn't have the thickenings present on the wall. It cannot sustain those thickenings, right? They cannot sustain those thickenings. So first answer becomes wrong for that matter. Okay, it cannot be right. This is not common thing. Okay. Now, second option, option B, pores on lateral walls. Okay. Now, understand one thing. Your vessel elements, they are more pro in conduction task. Okay. So in vessels, you will find pores on the lateral wall. Let's say if this is a vessel I'm drawing. Okay. If this is one vessel element I'm drawing, then you will find pores on the lateral walls. You will also find pores on the end walls. Okay, you do find pores on the end walls as well. But lateral walls do not have pores in case of sieve tube elements. Again, sieve tube elements are not having pores on the lateral wall. This is again a very major difference between both the conducting elements. Okay, when you talk about vessels and when you talk about sieve tubes, then this is one of the major differentiation you put forward. Okay, so again, this is a wrong answer. Okay, option C, presence of P protein. This is straight away wrong because vessels do not have P protein. Okay, vessels do not have P protein. Okay, P protein is present in sieve tube. It is responsible for plugging. So what do we left with? We are left with option D, which is right. Now, understand one thing. Vessels, okay, and sieve tubes, both of them are enucleated. Both of them are enucleated. But vessels are dead. Okay, they are dead parts. Sieve tubes are live. So if I say that both of them are enucleated, how can sieve tube survive? How can sieve tube survive without nucleus? Rebecca, one is dead, the other one is live. Okay, so if vessels are dead, there is no nucleus, it is cool for them. Okay, it is cool for them, right? No problem. But what about the other one? So I got an answer from Meenakshi. Meenakshi, very good. Sieve tubes are live. They are getting their metabolic requirements done from the companion cells, right? That is why they are companion, always helping them out. Okay, that is why they are getting their requirements fulfilled by this thing. Okay, so this is the only commonality between both of them. Okay, understandable? This function, this particular concept is understandable to all of you, right? Okay, moving on to the next question. Right. So this question is about some identification. Okay, exactly. This is asking some sort of identity. Okay. It says that suppose if you are provided with an old piece of dicot stem and a dicot root, okay, how are you going to differentiate between the two? How would you say that, okay, this is stem and this is root? Which anatomical structure you will have to look into? Which thing will you look into? This is, I'm not going to explain the particular options here because it's a very easy question, right? Anyone? Okay, I got an answer from Vaishu. Okay, Rebecca. Protoxylem. This is right. Okay. Why? Because you find that protoxylem plays a role over here. Okay. Protoxylem has a role over here. Okay. In case of stem, okay. In case of stem, you find that protoxylem is in dark. Okay. In case of stem, the protoxylem is in dark. In case of roots, it is exarch. Very good. Okay. And dark simply means that protoxylem is present towards the center, towards the pith. Okay. Towards the center. And in exarch, it simply means that protoxylem is towards the periphery. Okay. Towards the periphery. Hi, Raj. Hello. Welcome. Understand? So both of these features are very distinguished from each other. You can simply look into the protoxylem and mark a particular structure as stem and another structure as root. As simple as that. Fine, everyone? Understandable? This thing is understandable, guys? Okay, next question quickly. Passage cells are thin-walled cells found in. Now, we know that passage cells will always come across when you are studying a structure called as endodermis. Okay, always remember passage cells are something associated with endodermis. So I've given you a hint over here. Okay, right? So when you will read into the options, you'll find option number B telling you that endodermis of roots facilitating rapid transport of water from cortex to pericycle. Okay, why? Because endodermis, if you pay attention, endodermis of roots has something called Casparian strips. Okay, endodermis of roots is blessed with Casparian strips. Okay, and Casparian strips are made up of lignin and suberin. And both of them do not allow movement of water via the cell wall. Apoplastic movement of water cannot happen. Exactly. Okay. That is why we have certain cells who do not have lignin and suberin. Passage cells do not have that. Okay. They do not have that. So B is the right answer. Exactly, Meenakshi. So passage cells are basically allowing the directed movement. Okay. Directional movement of water directly to the xylem. Okay. Giving them a kind of a, guideline. Okay. Giving them a guide to move to the xylem. Okay, very fine. I can see many of you already having the right answer at your end. Very fine. Next question. This is an assertion reason question. We'll do it in the assertion reason itself. Okay. Assertion is that monocot stem has collateral open vascular bundle. Now, assertion is wrong in the sense you do not find open vascular bundle in monocots. Open means that cambium is present. Okay, open means that cambium is present, which is wrong. You do not have cambium in monocot. They do not show you secondary growth. So this is a wrong assertion. Assertion is wrong for that matter. Okay, coming on to the reason, open vascular bundles is without vascular cambium. Who says that? No, open vascular bundles are not without vascular cambium. They are with vascular cambium. Okay, so as a matter of fact, both of them are wrong. Okay, both of them are wrong. As simple as that. Exactly. Okay, So we arrive at option number D, which says both assertion and reason are false. Okay, just remember, if it is closed, it means no cambium. Okay, if it is open, it means there is cambium. Okay, cambium present. So this is a key, and you do not find cambium in the monocots. Okay, very good. Next question. Root cambium is derived from. Okay, root cambium, which is resulting in the secondary growth of root. Okay, that results in secondary growth of dicot root. Okay, it's derived from. Okay, now you understand that in case of roots, the derivation of root cambium is entirely occurring from the secondary meristem. Okay, there is no involvement of primary meristem. Okay, it is entirely made up of the parenchymatous tissue. They get meristematic and they form a star shape meristem. Okay, they form a star shape meristem. Very good. So it's not primary, it's not intercalary, it cannot be apical, it is secondary meristem. Okay, they form a star shape meristem over there. Okay, very fine, very good. Next question. Porous wood contains. Right now, this is again a very good conceptual question that came in AIMS. Right, porous wood, the name implies that you have to talk about one particular feature, which is why it is getting the name porous. Okay, and vessels, for that matter, is something which is making the wood porous. Why? Because vessels have wide lumen. Okay, they have wide lumen. You can see in the particular diagram I'm making, this is such a wide lumen, and that appears like pore when you are looking into the microscopic structure. They appear like pore. Okay, and porous wood is a characteristic feature of angiosperms. Okay, the flowering plants, for that matter, you do not find such type of wood in the other types of plants, right? And you can have, on that matter, you can have something called ring porous. Okay, you can have something called diffuse porous, right? And this remains your homework. Ring porous and diffuse porous, I have given you two terms. After this particular session, you will have to find out what exactly is ring porous and what exactly is diffuse porous. Write it down somewhere. This is going to go with you, and you will have to look up to the ring porous thing and the diffuse porous thing. Okay, interesting definition you'll come across, I'm sure. Okay, exactly. And the vessel is the characteristic of angiosperms, right? So we do have certain exceptions as well. I'll let you know about that. Okay, next question. Which of the following statement is true? Okay, so we have to read the statement and find out which one is true. Okay, option number A says that vessels are multicellular and wide lumen. Now this appears to me as right, because vessels are multicellular. They are made up of many vessel elements. Okay, they are made up of many vessel members, and they do have a wide lumen. So this is appearing to be true, right? And this is going to be the answer, right? Okay, because if this is a multiple choice question, one has to be right. Very good. Others are not right. Why? Tracheids are multicellular. Wrong. Tracheids are not multicellular. Okay. Vessels are unicellular. Wrong. They are not unicellular. They are made up of many vessel members. Tracheids are unicellular. That is right, but they do not have wide lumen. They have narrow lumen. Okay, which is why all the other options are wrong and the first one is right. Okay, so I guess this is understandable. And coming on to the last question of this session, which happens to be assertion and reason again. Okay, so we'll read both of them, try to find out which one is right, which one is not. Okay, in collateral vascular bundles, okay, phloem is situated towards the inner side. Now there is a problem. In collateral vascular bundle, okay, you do not find phloem inside. Okay, if I happen to draw the vascular bundle collateral, okay, something like that individually, okay, and this portion is where you have the phloem present, and this is where you have the xylem present. Okay, and this is, suppose, let's say pith over here. So I'm just drawing a section. So phloem is not towards the inner side. Phloem is actually towards the outer side. Okay, so this is a wrong assertion. Just mark it right here. Okay, this is a better way of doing assertion and reason. Okay, yes, many people are giving me right answer. I'm so happy. In monocot stem, cambium is present. No, this is false. Cambium cannot be there. This is a wrong reason. Okay, so D is right. Very good, very fine. So from these answers of yours, exactly, Meghna, there is no cambium. Reason is wrong, as well as assertion. I can see many of you have a good concept of anatomy. Keep it up. Okay, so with that, we finish our session on this particular topic. Okay, you can expect certain questions in your examination, because or however it hard it appears, it's not difficult to retain them. Okay, the concepts are retainable. Okay, so I guess this particular section, this particular topic we have discussed is of help to you. Okay, it has helped you to revise some of your concepts. You will definitely go back and look into some of these. Okay, open your book, try to find out. Okay, so thank you everyone for being here. Thanks for attending the session, being actively participating. See you in the next session. Till then, all of you be in good health. Keep studying. Okay, have you enjoy your life. Bye bye. All right, sure. Okay, Nivya, sure, of course. Bye everyone. Take care.